Hello, so my name is Maria Rojas and I will present the content of this paper here. Okay, so we got a problem, and the problem relies on the large availability of protein sequences, and not only this, but also in the lack of experimentally validated function that we have for most of them. Okay, so this is a very difficult problem to solve, since there are lots of genomes coming and more are coming, and the approach that people are usually taking is the homology-based one, which is a strategy that has been working for model organisms and sister species, but we know that for non-model organisms it's a difficult method to use. In fact, when we look at the percentage of annotated genes in the model species, we have still a lack of annotation, functional annotation in a handful set of genes, like for instance around 18% in fly and 28% in the worm. So when we move to the non-model organisms, what we have here is even less information, since we can only annotate half of their genomes. Okay, so we decided to use other methods based in artificial intelligence or machine learning, whatever you want to call it, okay? And at two levels. First, we would like to know how well do they perform at the organism level, not using averages of data sets that contains information from all the species, and if we can use these methods in order to annotate them here, okay, in this level of uncertainty, which is the non-model things. To this regard, what we are doing is we are using two different methods, the protein language models and convolutional networks, okay? And for the first flavor, which is the organism calibration, then we are using the Goa UniProt KV as a standard. However, given the lack of annotations in the unknown sequences, what we are using as a reference is the EGNOG mapping, which is based on homology-based and phylogeny-aware. So we can see that in the case of the model organisms, here I will show everything for the worm, we have an increased annotation coverage. You can see here the orange bars are the real Go annotations from the proteome of this worm, and you can see that the language models here in green and the convolutional networks in gray are providing more coverage in the number of proteins, are also in the number of annotations. Okay, what we can see here is that also they guess the real Go term. Okay, so this is an hypothetical schematic view of a Go graph, and a hit will be when they guess the term. Okay, and we can see here, the darker the better, that the language models here at the right, the columns at the right for each category, are providing more real annotations, more guessed annotations than the other ones. In this particular case, if we removed the annotations obtained by hidden Markov models, what we can see here is that the language models still provide more annotations. So looking at the information content, which is giving an idea about the detail of the annotation, we can see here in green the language models are doing much better than the convolutional networks. In the semantic similarity, we know that the molecular function is the category that is better predicted by language models and by any other method, okay? And this performance is going low when we are moving through the cellular component and the biological process categories. Another experiment that we did is the ability to recover functional information from transcriptomic experiments. So we did that, and here I'm showing one experiment from the worm, in which you can see here the downregulated function. The top lines are the language models. They are recovering functional information if we are using the predictions at larger semantic similarity values as compared to the other convolutional networks methods. Okay, so basically language models are outperforming convolutional networks. Now the idea is, how do they annotate at this level of uncertainty? And what we have seen by annotating 24 million of genes in almost 1,000 animals, what we have seen is an increase on the coverage of annotations and in the coverage of proteins annotated, as you can see here in this graph. And here I'm showing you the biological process information content as expected, and as we have seen for the model organisms. We know that the language models are providing more detailed information, larger IC values, as compared to the convolutional networks. And the semantic similarity in this case is a little bit misleading, since it is using half of the genome, which is obtained by homology. So Which kind of insights can we get from this effort? Well, we can get insights from intrinsically disordered proteins. For instance, we know that the intrinsically disordered proteins, the more disorder, the worse their annotations are, okay? And this is one gene that belongs to fly, one of these unannotated genes, which is an intrinsically disordered protein. And the language models are providing these terms, like meiotic cell cycles, non-transduction, and so on. And we know by transcriptomic experiments that this gene is expressed in the somatic cell of just these male germ cell lines, okay? So it's coherent what we are getting with the expression data which are available. Another way is to look at the insights for proteins with no homologs. We have here the tardigrade, and then we have this overrepresentation of terms that are related to the resistance of this guy, okay? So we have the cellular response, cellular response to heat, and so on. So these terms are associated to genes, and having these genes that have no homologs anywhere else, we can get them and then run certain experiments. So basically, to finish, we have also created this pipeline. The pipeline is a bioinformatics pipeline, which is implementing the prototype 5 of Burka-Ross, the GoPresim method, and we are doing additional GoTerm analysis. And then the only input that you need to get is your protein sequence file, and then you will have your Go annotation file. Here you have the QR code. And that's it. So thank you so much. If you have any other question, you can go to the presentation and to go to the poster, which has more detailed information. And we want to thank also the language, well, the funding bodies, okay, and people who is providing the icons. So thank you.